Good afternoon, everyone. Welcome to another online lessons. In this video lecture, we will be talking about the B cells activation and crosstalk between the T helper and the B cells. As you know, in the previous lessons, we've been talking about the T cytotoxic T cells and the T helper cells with the MSC class 1 and MSC class 2, and describe the T cells receptor as well. So we want to know the interactions between the B cells and the T cells and how the immune system get activated when these interactions would happen. Okay, remember one thing that always what we talked about, the similar logical lecture I told. In the previous videos, we've been talking about the T cells, and we know the activation of T helper cell is one of the major events that will help in uplifting both cellular mediated immunity as well as the humoral mode of immunity. Basically, B cells are responsible for the humoral side of immunity because B cell have the capability of converting itself what is known as a plasma membrane or plasma cell, sorry. Uh, the the humoral immunity, if you talk about the humoral immunity involves leukocytes called B lymphocytes. The B lymphocytes are produced and mature in bone and are usually found in lymph nodes. So during the innate immune response, macrophage has ability to basically to engulf the pathogens. Well, in the pathogens we basically differentiated in two types. One of them could be exogenous pathogens like the bacteria, parasite, virus, or the another kind which it could be the endogenous pathogens like the virus or the cancer. The B cells are a type of lymphocyte, yeah, that's the reality, that have an essential role in the immune systems. The goal of these types of cells is to make antibodies against pathogens. So, if you want to talk about the functions of the lymphocytes, B lymphocytes, the functions of the B lymphocytes is to produce a variety of the antibodies that will interact with the antigens. And we've got different kinds of antigens. We've got bacteria, we've got virus, we've got parasites, we've got cancer as well. So if you want to say the B lymphocytes, so it belongs to adaptive immunity, and it is antigen specificity. Why it's antigen specificity? Because it's produced basically the bone. It's produced in antibodies, and these antibodies interact with the antigen. And the humoral immunity, yeah, because it's a part of the humoral immunity, and the raw format in the bone marrow of mammals and often transfer to the spleen. The activity of this type of cell can be described as a humoral response. And another, but the different type of the immune response is the cell-mediated immunity response. The cell-mediated immune response is largely controlled by the T cells, okay, by the T cells, and the B cells are quite different than the T cells because basically B cells release or produce a variety of the antibodies. These antibodies interact with the antigen, but it needs to combine with the T cells or interact with the T cell, specifically the T helper cells, to activate the immune system. After that will lead to release a lot of plasma cells because the final destination for the B cells is plasma cells. Okay, here the lymphatic systems or the lymphoid tissues. So the lymphatic system is a network of tissue and organ that help protect the body from toxins, waste, and the other unwanted material. Primary functions of the lymphatic system is to transport lymph and the fluid containing infection, fighting while the blood cells like the neutrophil or the monocytes or the macrophage throughout the body. We've got the primary lymphoid tissues are sites where the lymphocytes let's turn on the laser. Basically, well, are sites where lymphocytes developed from the progenitor cells into functional and mature lymphocytes. The other primary lymphoid tissue is thymus. So the first one is the primary, is a bone marrow, because where the lymphocytes developed into the functions and get matured. Okay, the secondary on the other primary lymphoid tissue is the thymus, site where the progenitor cells from the marrow differentiated into mature thyme. And we've got the lymph nodes and spleen and tonsils and lymphoid tissue within GIT and respiratory tract. Okay, here I'm talking about the B cells development and maturations. So the first thing I will start talking about is the hematopoietic stem cells, which is the multipotential, the hematopoietic stem cells that produce to other blood cells. So basically this process occurs in the red bone marrow and core and of the most bones. And then the process by which all mature blood cells are produced, and it balance numerous productions. It needs, the average person produces more than maybe 500 billion blood cells every day, with the need to regulate the number of each blood cell type, the circulation in vertebrate. The vast majority of the hematopoiesis occur in the bone marrow and derive from a limited number of the hematopoietic stem cells that are multipotent, are capable of extensive self-renewal. So let's talk about the hematopoietic wall divided to common myeloid and the common lymphoid. The common myeloid is divided into the basically to the thrombocytes, erythrocyte and mast cells, and the myoblasts were divided to basophil and the neutrophil, with the eosinophil and the monocytes. Basically, if we want to talk about we know that we can collect the blood from the healthy donor and then separate the basophil and the neutrophil with the monocytes, and then the monocytes will develop to the macrophage, these in here. And basically with the same things with the lymphoid progenitor, which is the small lymphocyte. It's the B cells with the B lymphocyte that will produce a variety or many kinds of the antibodies. With the T lymphocytes, T lymphocytes will be divided into basically CD4 and the CD8. The CD4 is T helper T cells, and the CD8 is the CD cytotoxic T cells. And we've got the natural killer cells. The natural killer cells is a part of the basically of the lymphocyte of the T lymphocytes. It works like the CD8, like the cytotoxic, because it's, as I mentioned before, it works. It's release the granular cytokines that will lead to distract and damage the cells by making pores inside the cells that will lead it to induce apoptosis. So we've, if you remember, in the previous lessons, I've been talking about the basically about the basophil with the, and we said the basophil release the histamine, which causes dilation of blood vessels in the infected area, causes the redness in the capillaries in infected area, also become more leaky and causes the edema. We, as well as the neutrophil as well, these cells are recruited to the infected area and engulf pathogens that will enhance the immune systems by releasing cytokines, granules, and initiation of the oxidative burst. That means it's the each neutrophil could be capable of killing 20 bacteria before dying. We've got the eosinophil as well. In eosinophil, these cells are specialized to fight certain parasites. That the eosinophil is a defense against helminth's parasites. So eosinophil works on the parasites. As we've got as well the monocytes, and the monocyte that will develop. So it could be to not just to basically to the macrophage, but also it could be developed to the dendritic cells, and the dendritic cells, well, in the macrophage and the antigen and the dendritic cells is a part of the antigen presenting cells. So this is the process of antigen peptides on the cell surface and then loaded on the MSC class 1, as I mentioned before, including as well the B lymphocytes. So the macrophage is like multiple immune functions, including detections, phagocytosis of pathogens, the antigen presentation to the T cells, and the cytokines productions. The macrophage basically it's come from circulating monocytes, and then the monocyte will some stimulating factors will lead to when they circulated in the blood as immature form, and then when it gets the tissues, that will lead to differentiated to mature macrophage. Then the macrophage, it has the ability to engulf the pathogens and distractions, and after that will display on the cell surface. That's the same way with the dendritic cells as well, because we've got two forms, as I mentioned before. We've got immature and we've got mature, and then both of them has the ability to engulf the pathogens and express it on the cell surface. That will enhance the activated or illustrated the immune systems. That's like the brief explanations, all the diagrams about the hematopoietic stem cells, and divided, as I mentioned, to myeloid and lymphoid, and we've got them different kinds of the cells, and we can in the basically in the lab we can just take the blood from the healthy donor and then isolated the all kind of the cells. So basically, the B cells works as immune security and in the body in search of foreign and concerning particles. It could be any kind of the antigens, and it works as immune encounter. This types of cells finds a foreign object and becomes activated and can divide into either plasma cells or memory cells. I will explain that later, the functions of the B cells and maturations. So let's give you an overview of the B cells development. B cells basically are generated in the bone marrow, and takes 1 to 2 weeks to develop from the hematopoietic stem cells to mature B cells, and the sequence of expressions on the cell surface. So because of the B cells, it's look like the T cells. It has the receptor. This receptors, it's like the it's tightly connected to the cell surface of the B cells, and with with the antibodies that's expressed on the cell surface. That's allowed to differentiate it of the B cells proliferations at the various stage. The movement within a bone marrow microenvironment, immature B cells, basically the early B cells development constitutes the steps that lead the B cells and expressions of the surface, mean the immunoglobulin productions of mature B cells, and mature B cells leave the bone marrow and migrated to secondary lymphoid tissues. B cells that interact with exogenous antigen or T helper cells, which is it's mean the antigen independent phase. So the development of the B cells, sorry, it's differentiated of the B cells occurs in the bone marrow through the life and of individuals. Basically can recognize the B cells like we've got the pro B cells, that mean the before get matured, and we've got the pre B cells, that mean the after maturations. So immature B cells, the cells which express the immunoglobulin that's on the cell surface, so special kind of the immunoglobulin. It's not just the whole immunoglobulin, because I think you go to listen about the antibodies, and you know the antibody, it's consisted from the IgG, IgM, IgE, IgD, IgA. So that's the when the immature B cells leave the bone marrow, that will lead to release one kind of the immunoglobulin, which is called, or express one of the immunoglobulin, which is called IgM on its surface. And the immune system must create it like the it has a capable of detections the large variety of the antigens when on when the antibodies expressed on the B cells. So let's explain that. So you know the bone marrow, it's basically it developed from the hematopoietic stem cells. So in the stem cells, to be mature, then the wool leaves the bone marrow undergoes the further differentiations. So we've got the two kinds of the B cells. We've got the immature B cells and mature B cells. In immature B cells, that means express one kind of the immunoglobulin. But the mature B cells which lead to express the IgM or the IgD on the cell surface. That it means that another kinds of the immunoglobulin to express on the cell surface of the B cells. Well, we know the activations of the D cells, it leads to proliferations into plasma cells, and then it could be that lead to release a lots of variety of the antibodies, will lead to activated the immune systems. Could be activated by itself, or it could be activated by connected with the T helper cells. Okay, so let's jump on the another. Okay, so basically we've got two kinds of the cells, the cellular immunities and the humoral immunities. And the humoral immunities, that's it means the antibodies or produce a variety of the antibodies, because it's the humoral. If you look into the humoral immunity and the cellular immunity, so well, it's that the means when the infections begins, the innate immune system immediately use the non-specific mechanisms and inflammation to destroy invading pathogens. The adaptive immune systems is then activated after the innate immunity, and that leads to the it's not general actions, but it needs several dates to activate, and then the specific defense mechanisms of the adaptive immunity. Basically, so the adaptive immunity, it could be divided into the cellular immunity and the humoral immunity. Well, if you started with the cellular immunity, so we've got like here in the T cells, that is it either the T helper cells or the T cytotoxic. Here we give an examples on the T helper cells, and the T cells has a receptor which is called the T cell receptor. The T helper cells cannot be activated without connected with the fragments peptides on the cell surface of the MSC class 1 that will express it on the antigen presenting cells. We've got here the example as the pathogens like the intracellular microorganisms like the virus. So when the virus gets into the site of the infected cells or into antigen presenting cells, where in the antigen presenting cells it could be macrophage, it could be dendritic cells, or it could be the B cells as well. But here we've got two types, the macrophage or the dendritic cells. So then the macroorganisms will get into the site of the cells. That will lead, there is the phagolysosomes, that will lead to distracted the antigens into fragments. These fragments will express on the surface of the MSC class 2. Then the fragments would bind with the T cell receptor on the cell surface of the T helper cells. This is a process will lead to activated and release some cytokines and lead to the proliferations and activation of the T helper cells, that the clonal, it's called the clonal T helper cells. At the same time, that will lead it to call the C cytotoxic T cells of the CD8, because it has the granular toxins will release to kill all the foreign microorganisms, and then they call the macrophage to engulf the foreign body. This here, the killing of infected cells, that means it's induced apoptosis. Okay, so here we've got, so that means that this involves the leukocyte, which called the T lymphocyte, and they produce it, and the T lymphocyte produces in bone marrow but mature in thymus, and the helper T cells release the interleukins and the interferons. The cytokines which is released from the T helper cells, it's the cytokines, interferons, or interleukin. T lymphocyte, express it on the cell surface like the receptor, which is called the T cell receptor. And this with the cytotoxic T cells here, basically is migrated to the infected area and binding to the specific antigen, and then in addition the lymphocytes can from the memory cells or suppressor T cells, that means to regulate the immune systems. In the humoral immunity, we've got that, the B lymphocyte expresses the receptor on the cell surface, and that the receptors would tightly bind to the with the like the antibodies or the immunoglobulins, and then the B cells, when expressed on the cell surface of the B cells, they bind to these macrophage bound antigens, and the lymphocytes can bind to the specific antigens, and then that lead to the it could be there is the interactions between the B cell, the B lymphocyte, and B T helper cells. But here I want to just describe it that the B lymphocytes can release or express a variety of the antibody on the cell surface. It depends on the pathogens, so it cannot just express just the many kinds of the antibody. It's need no to express like these antibodies, it basically surrounding the B cell and engage interactions on any sets of the pathogens and load it on the MSC class 1 if it has the, because it's kind of the basically it's B cells is kind of the antigen presenting cells. So the T helper with basically here that's the B lymphocytes will lead to release many kinds of the antibodies. It can recognize it the antigens, because there are the antibody, it has the variable regions and the constant regions, and variable regions that will lead to interact with the pathogens. And when mutation going on, the variable region called these variable regions called the somatic somatic mutations due to the maturations. They have the different types of the structures, can many with the many variety of the antigens, right? So, and these antigens, it could be activated, lead to well, basically to activate the which is called the opsonizations, and the process which is called the opsonizations, all that lead to engulf the pathogens by the macrophage and use it the phagocytosis that lyses, and then attach it to the complement. In the complement, when attached to the complements, that will lead it to enhance the immune systems by by the process which is called the phagocytosis. The phagocytosis could be by the macrophage or could be the dendritic cells that will lead it to release that will lead to engulf these the pathogens on the peptide fragments that attach it to the variable regions on the antibodies and engulf it by the phagocyte or the macrophage, will lead it to distractions, and then the totally lead the totally damage. And then basically the antibody, if it was for instance IgM or IgG, so the case they will produce a lot of IgG. If it's IgM, then should produce it a lot of the IgM. So that's, it depends on the types of the antibody to recognize a specific type of the antigen. Okay, as I mentioned before, the functions of the B cells, it could be like the immune security or immune counter. So the B cells patrol the body in search of foreign microorganisms or foreign bodies or any pathogenic particles or that could be harm the immune systems. The immune systems cannot recognize it, all the pathogens. They to interact just with the antigenic epitopes, which is like the part of the antigens, as I mentioned before. It could be the polysaccharide or it could be the peptide or nucleic acid, and using that the immunoglobulin receptor, because the B cells, it has the special receptor on the cell surface, that's which is titled, which is called the immunoglobulin, to present the antigenic peptides. That when they present it, that will a peptide it will produces to or presented to the T cells. Consequence up to neutralizations and process of the original antigens. The B cells as well, it's responsible for the humoral arm of the adaptive immune systems, because it's a part of the mediated immunity. It's called the humoral, because the B lymphocytes undergoes many cycle of mutations that will lead it to release the many kinds of the antibodies and the specific antibodies that expression basically to the original antigen expressed on the B lymphocyte. The IG, it means the immunoglobulin bounded to the conjugate antigens. That's mean it's that's mean on the surface of the B cells, we've got the immunoglobulin, and any kind of the immunoglobulin, which could be the IgM or IgA or IgA or E or D, that will attach with the or connect with, or well, that interact with the antigens. And when the antigen is bound to the antibody on the cell surface of the B cell, that will send the signal to inside the B cells. So for instance, if the pathogen matches the antibody, for instance, this is the IgM release on the cell surface of the B cells. So that's antigens will bind to the IgM and then sends the signals to the inside to the B cell to release more antibodies of the same types, which is the IgM. But if wasn't the IgM, but the IgM was expressed on the cell surface, that will lead to so many mutation or somatic hypermutation inside the B cells, which lead to release the another kinds of the antibody expressed on the cell surface of the B cells, which interact with the antigens. In this case, will lead it to activated the complement systems. Why the complement system? Because basically the antigens, when it's released, that will lead it to interact with the complement, and the three, we've go to the three subcomplement of the complement will be activated, and we've got the B cells, the certain antibody expressed on the cell surface, which is known IgM or IgG, can bind it to the complement, and then when bind it to the complement, that will lead them release the vasoactive mediators such as the histamine, for instance, and then the complement attach it to the microbial pathogen. The microbial pathogens attach it to the variable regions of the IgM or the IgG, and then lead it to the activated the macrophage or the neutrophil to interact it with the complement, and then will lead it to these lead it to elicit or boost the immune cells. Basically, the complement systems, also play a clear role in the cell hemostasis by contributing lysis of the cells undergoing apoptosis, and especially the extracellular plasma membrane change occurring during apoptosis, which allows cells to bind the complement molecule. We've got a lot of complement, like the complement 1q, as I mentioned before, that will help to the phagocytosis, taking up the antigen. So B cell activations occurs in the peripheral lymph organs, and the activation of the B cells leads to proliferation, differentiated into plasma cells and into the memory cells as well. So we've go to the plasma cells at the end stage of the B cells differentiated, and occurs in the specialized region of secondary lymphoid organs, spleen, or lymph nodes and germ cells, but as well B cells get activated by the antigens and the class of the isotype switching. The memory, basically, if you talk about the plasma cells, so the plasma cells is plasma B cells are a group of the cells that divided after activation. Okay, so divided after activation, or after when the B cells get matured, and then the following the contact with the foreign body. So mean when the B cells will get activated and then express the immunoglobulin on the cell surface, then the antigens attached to the antibodies will lead to release a lot of the plasma membrane that will be activated. This is the final destination for the B cells, which is get the plasma be the plasma cells, and the antibodies will help to kill the foreign invader, and the antibody will attach to the bacteria or the virus and allow to the activations of the complement systems and the phagocytosis. In following the immune response, these are quickly destroyed. So basically that is the plasma membrane. But if you talk about the memory cells, so that is seen in the T cells dependent immune response and survive for a long time because are a class of the B cells. They divided after activations, and the activations occur following the infections. Following the first of initial response, an antigen clone of B cells are formed. Most cells are plasma cells, but some become memory cells, which in well basically the memory cells remain after infection has been resolved, and following the subsequent exposure to the same antigen. So if you get the same exposure, that will be, we've got already the memory cells in your body that's already in a place to protect the fight and the current infections. An additional clonal will be response to the second attack, and more antigen protections will produce. So that means the B cells activations will be divided in two types. This is the plasma cells and the memory cells. We've got the others kind of the during the B cells activations, but these are important kind of cells, which is the plasma cells and the memory cells. So the memory cells could be ready when the cell to protect the body when you get the reinfected again with any kind of the infection. So you've got some of the memory cells in these cells, and there will be response straight away when you get the reinfected. In the plasma cell, that's mean the final destination of the B cells will release the plasma cells, and the plasma cells will release a variety of the antibodies that will lead it to bind to the complement and induce its opsonization that will enhance to engulf the pathogens. So, development of the humoral immune response. A humoral immune response refers to the protections of secreted variety of the antibodies by the plasma cells. As I mentioned before, the final destination for the B cells is the plasma cells following antigenic stimulation of the B cells. So, the B lymphocyte then undergoes the many cycles of mitosis to produce the plasma cells and the memory cells. The plasma cells, it has ability, or it's capable to produce antibodies specific to the specific antigen. Then that they're exposed to the lymphocytes. So the antibodies expressed on the cell surface of the B cells that the closely bound to the B cell receptors, and the variable fragments of the antibodies will bind to the antigens and lead to activated the B cells, and then will release and divided and differentiated into plasma cells and the memory cells. We've got here the primary and secondary immune response. So the primary immune response refers to any immune response of the immune systems that include the protections of the antibody or the cell-mediated immunity, and it's occur following the first exposure to the foreign antigens. So basically, you've got the exogenous antigens. It could be bacteria or virus or parasite that's within individuals that lead to activate when it gets inside the body, that the immune system recognizes that there is a foreign body and then activated the humoral response. The humoral response, that means the B cells will be activated. The B cells, it has the B cell receptor on the cell surface and closely bound to the antibody, and then the antibody, it consists of from two regions, like the variable regions and the constant region. Variable regions antigens will attach to the pathogens or the antigen. That will lead to send a signal to the inside the B cells, and the B cells will be activated and then will be converted to the plasma cells and the memory cells. And then, the kinetic of the primary response as measured by the serum, the antibody level, it depending on the nature of the antigens, because the antigens, its route of the administration is the presence or absence of adjuvant. So adjuvant, it's a substance that enhances the body immune response because it increases the immunogenicity of the antigen ability to produce immune response. When it's injected to inside the human body, that it depends on the many factors like the size of the antibody, dose of antigen, of the biochemical composition. Some antigens are more immunogenic than the others, so when injected the animals with the antigen and adjuvants, the immunogenicity is like the directly increased, and the many vaccine which are inactivated antigenic preparations and mix it with adjuvants, because and sometimes if you we've got the solutions of the material, it has the some kinds of the like the this could be the gram-negative bacteria or gram-positive bacteria, and that lead activated the immune systems by expressed of the MSC class 1 on the MSC class 2. The second type of the immune response is secondary immune response, and the secondary immune response refers to any immune response of the immune system that occurs in response to the subsequent exposure to the particle antigens. So second exposure may occur even after a year. That means the activation of the memory cells by the antigen. So the B cells could be activated to the plasma cells and the memory cells, as I mentioned before. So the plasma cells, that lead to release the variety of the antibodies, but the memory cells will stay in the cells and will be activated, could be 1 year after they're reinfected with another kind of the antigen. And the secondary, it could be the same kind of antigen, but that will know the immune system. There is some foreign bodies will invade the or attack the human bodies. The secondary response has a short lag period, reaches a greater magnitude and lasts longer. The secondary response also is characterized by secretion, that means the antibodies with a high affinity for the antigens. The isotype other than the IgM, because it's predominant and the second exposure to the same antigen, and basically result in immediate production of productive antibody, mainly IgG, but may see some IgM as well, because the IgM, it's already found in the if you found in our body. If you want to differences between like the immune between the primary and secondary immune systems, that's which is the both of them, it's so the primary immune response occurs in response to the primary contact with the antigens. That means it comes from the outside of the body, whereas the secondary immune response occurs in the response to the second and subsequent exposure to the same antigen. So that is the differences, because you've already got the memory cells that will let your body know there is some foreign body. So that will, so you infected already with this kind of the antigen, that will lead to distract the same antigens, because you've already infected with this kind of the antigens, because it's a response to the second and subsequent exposure to the same antigen. So, B cells activation by many antigens requires both binding of the antigens by the B cell surface immunoglobulin and the B cell receptor, and interactions of the B cells with the antigen specific helper T cells. The helper T cells recognize the peptide fragments derived from the antigens internalized by the B cells and displayed the B cells as a peptide MSC class II complex on the T helper cells. So basically, as you know, so if you go to the B cells, the B cells cannot be activated. It could be activated, but for just for the short life, and in the absence of the T cells, because there is a connection between the B cells and especially the T helper cells. So you know the B cells, it has a receptor on the cell surface, and this the receptors are tightly or tightly bound to the antibodies, and the antibodies will get the signals from the outside of the cells, and that lead to presented, because the B cells is one of the antigen presenting cells, so that means it has the MSC class 2 expressed on the cell surface. The MSC class 2 activated the T helper cells. The T helper cells, it cannot recognize the peptide fragments until express or display on MSC class 2. So that when the pathogens expressed on the MSC class 2, that's can the T cells recognize the T helper cells can recognize the concerning particles that's expressed on the MSC class 2. So if it's the maturation of the B cells happened in absence of the T cells, so it could be happened with that rapidly matured into short term life the plasma cells without undergoing to the somatic hypermutation or the class switching. So you know the class switching or the somatic hypermutation, that's mean if it's the antigens that's the B cells can recognize it on the cell surface. For instance, if it's the pathogens doesn't match with the IgM, that will lead to set the signal to inside the B cells and see the B cells. It doesn't match me, so we need to produce it another kind of the antibody that will lead it, for instance, the IgG, and that lead to the somatic hypermutation, or it's mean the class switching. That's mean the IgM switch with the instead of the IgA, and secreted the IgM antibody but with the low affinity. Do not contribute it. It doesn't have the memory cells, because you know the activation of the T cells, it will be divided to two kinds of the cells, the plasma cells and the memory cells. And the memory cells, that mean when they infected with the same antigens, but the plasma cells, that means it when the cells it's produced many types of the antibodies. We've got the B cells maybe professionally follow this no follicle differentiation pathway, as they appear to be much less dependent on the T cell help for the antibody productions, because if there is no T helper cells, so they cannot the B cells cannot be activated and release a variety of the antibody and produce the plasma cells. So here we need to discuss the B cells immunodeficiency. As you know, if it's the B cells immunodeficiency, that's mean the deficiency in the humoral immune cells, which causes the disrupts in the humor immunity, which can lead it to immunodeficiency. So it can be mediated by insufficient number of the B cells or the plasma cells. So the B cells cannot have the ability to produce a variety of the antibodies. That is the first thing. Or could be that lead to the poor opsonization of bacteria. Is a part of the late stage of the adaptive immunity, and as mentioned before, that the pathogens and the other particles are marked by the IgG antibodies. These antibodies, it could interact with the Fc receptors on the macrophage and the neutrophil, resulting in the phagocytosis. So the complement 1 Q complex and can be interact with Fc regions of IgG and the IgM immunocomplements activations. The classical complement pathway and making the antigens with the complements and bind to the pathogen surface through the alternative complement pathway. I think I described that in the previous lessons about the opsonization and how opsonization will happen, and the recruitment infections with the encapsulated. So here we discussed the disease caused by B cells defects. As I mentioned before, so it defects in the number of the B cells maybe leads to poor opsonizations of bacteria, and here it could be the B cells deficiency leads to different types of disease. They are called X-linked gamma globulinemia and IgA deficiency or IgG subclasses deficiency or the immunodeficiency with increased IgM and common variable immunodeficiency. So there are many types of the diseases caused by the B cells defects. So we're going to discuss some of them and describe what does mean each of them in the next slide. So here basically one of the disease which is called the X-linked hypogammaglobulinemia. It's causes of the disease that's mutations of the genes called the button thyroid kinase, which encodes the tyrosine kinase and an important in signals transductions protein. So basically, you know, the kinase pathway, it's we've go to the different pathway, but you want to know the pathway for the tyrosine kinase. It's very important in signal transductions because the pathway of the tyrosine kinase, when they get the signaling molecules like the cytokines, there is a receptor. The receptor lead to the cellular activator and then the transcriptions factor and the cellular effects. That it means that is like the well, the signals molecules cytokines, for instance, when the cytokines it's on the cell surface of the cells, that will lead it to the receptors will recognize these cytokines, and the cytokines bind to the receptor. The receptor got dimers, and then it's phosphorylated, and after the get phosphorylations, then the lead to the cellular activation process. When you've got the defect in the B cells, that will lead it to this kind of the disease, which is called Barton thyroid kinase, or could be the virtual absence of the B cells. That's how we can diagnose this kind of the disease, through the virtual absence of the B cells and very low level of the immunoglobulins, any kind. So you know we've got 5 types of the immunoglobulin, which are called IgG, IgA, IgM, IgD, IgE. If you did the diagnosis for the patient which go to the X-linked hypogammaglobulinemia, so you can find very low or the levels of the immunoglobulins, and it could be any kinds of the immunoglobulins. So basically, as you know, the immune system's helps protect the body from the harmful substance called antigens. And examples of the antigens could include bacteria, virus, toxin, cancer cells, and foreign blood or tissue from another person or species. Within the immune system detects an antigen, it responds by producing a protein called antibodies that destroy the harmful substance. The immune system response to also involve a process called phagocytosis, which is mean engulf the pathogens and then distracted in small pieces, and that leads to release some cytokines and then activated the immune system. So during this process, certain white blood cells will basically get bigger and destroy the bacteria and other foreign substance, a protein called the complement. It could help in this process. And I mentioned that before, the variable regions of the antibodies. So you know the antibodies, it expresses on the B cells. The B cells and the variable regions of the B cells will attach with the antigens, and then the antigens, and then after binding to the or binding to the antibody, that will lead it to attach with the complement, and then after that the complement which activated the opsonizations. I talked about the opsonization, which is engulf the microorganisms and lead to distractions. So basically, and the immunodeficiency disorder may affect any part of the immune systems. These conditions occur when the special white blood cells, you know, the T lymphocyte or the B cells, or both, do not function properly, and or your body does not produce enough antibodies. So because you know the mature B cell are normal in number and morphology, but failed to differentiate it to plasma cells because of defective interactions between the T and B cells, and mostly caused by the T cell defect. This defect is thought to be related to decreased number or functions of the CD4 lymphocyte, or occasionally to increase the number of the CD8 T lymphocyte. And then it could be lead to the immunosuppressor and disorder due to the medicines that weaken the immune system, such as the cortisone, for instance. So that is this kind of the also the immunosuppressor. It's a common side effect of the chemotherapy given to treated cancer. So basically, these clinical manifestations like the pyogenic bacteria, or could be virus such as the Epstein-Barr virus or Giardia lambella protozoa, that is a kind of parasite infected the digestive systems, or the high incidence of the autoimmune disease like the hemolytic anemia or thrombocytopenia. So in this case, if you needed to cure this kinds of the immunodeficiency due to the B cells or the T cells defect, we can treat it by intravenous gamma globulin must be given for the protections. Okay, this is a B cells that when we are talking about when it's get mature, is producing a plasma membrane or antibody producing factory of our body, and that start producing antibodies. These antibodies are very specific against pathogens. It can go against pathogen, interact with pathogens, and it can cause several destruction measures to the pathogens, and it can help the pathogen to be degraded by phagocytosis, as mentioned before, by the other phagocytic cells such as the macrophage, by the process called the opsonizations. Antibodies also help in fixing the complement. There is our part of our system, such as the bacteria, for instance, producing membrane attack complex or creating pores membrane of bacteria. So all this thing can be done by this antibody. All this response should be activated because a B cell normally presented in active form, and its B cells receptors on the surface, like the T cell receptor, is unique receptor on the cell surface of the T cells. And the B cells have the unique capability. B cells can produce it, the antibodies, and this antibodies found attached to the B cell receptor tightly, and this antibody present on the surface of the B cell. They can bind specifically to antigen or the pathogens fragments. And the B cells is another type of the antigen presenting cell. So antigen presenting cells means cells can engulf pathogens or fragments of pathogens, break it down inside the cells process that the antigen and can load it, that the antigen on the cell surface of the antibody, and it can be showcases to the rest of the cells. So these antibodies surrounded by the T cell by B cells can engage interaction of the pathogen and loaded it on MSC class 2. Okay, can loaded this on MSC class 2. So we've got here the T helper cells engaging the interactions between the B cells and the T cells, and these interactions between the B cells and the T cells are very important. And one thing I should tell, very important, the concept is that all the time in our body there is a variety of the T cells can find a specific antigen, bind the antigens, and then the antigen particles come from the outside of the cells of the immune system, not to able to recognize them only. So there are two, as I mentioned before, the variable regions of the antibody and the constant regions. Basically, the constant regions, they can attach or bind it with the variety of the antigens. When MSC class 1 is local fragments interact with the T cell receptor, also some costimulatory molecules which is called expressed on the B cells, which called the CD40, and the CD40 ligands, the CD4 presented in the surface of the T cell receptor, and all antigen presenting cells. So it's engaged interaction with the B cells with the MSC class 1, and this the CD4 is to check these interactions. So like they handshake, and like the handshake, you are fine. Okay, so what you are telling me now, the T cells started releasing the interleukin 2 and 4 and 5, and it's a very important cytokines then engage properly, and some way modified happen inside the B cells to produce the antibody, and then started to produce the antibodies. When it started to produce the antibodies, that means it started to activate the immune systems. So here we've got this is the B cells, and here the T cells or the T helper cells. The T helper cells, or the if either cytotoxic or the helper cell, because there was an interactions between the B cells and the T helper cells. So the T helper cells have the receptor which is called the T cell receptors, and basically the structures of the T cell receptors. It could be alpha, beta, or the gamma, delta T cells. Okay, so we've got all the T cells. This is the receptor. The receptors will bind it with the peptide fragments that will come from the outside of the environment, like the exogenous particles were infected the body, included virus or bacteria or cancer or toxin. So the antigens, and here we go with the B cell, and the B cell, it has the B cell receptor and has the antibody, and the antibody is tightly bound to the B cell receptor. Okay, so this is the antigen. It comes from outside of the cells, and as I mentioned before, it could be any kind of the microorganisms. The antibody released from the B cells, it has the variable region, okay, and it has the constant region, and this is the we've called the Fab regions or the FC region. Basically, the antigens, when attached to the antibody, that will send the signal inside the cells to the B cells, to release like they engage properly, and some many modifications will happen to produce a variety of the antibody and started the antibody. This was always, and then the that when engulf the antigen inside the cells and then distracted in the small particles or small pieces, that will lead to express it on the MSC class 2, because the MSC class 2 represented on the antigen presenting cells or the professional antigen presenting cells. When the MSC class 1 display or we can find, which is loaded the antigen on the cell surface, that will interact with the T cell receptor, the T cells receptor that's expressed on the T helper cells. This is the first interaction, and it's very important. That's mean there's an interactions between the T cell receptor and the peptide fragments, which is loaded on MSC class 2. We've got another signals which is released from the B cell, which is called the CD40. CD40 is a marker expressed on the B cells, and the CD40, when it bound to the CD40 ligands expressed on the T helper cells. The T helper cells, it has the co-receptor, which is called the CD4 with the T helper cells, and the CD8 in the T cytotoxic T cells. That will lead to, just to double check, it's come here to just to see it's engage the interactions with the check the interactions. It's like the handshake. Then the T helper cells will release some cytokines, which is called the interleukin 2 or 4 or it's 5, and that's will when this releases in the like the on the cell surface, that will lead it to start to produces a variety of the antibodies. Okay, that's lead to activated the immune systems or the antibody response. It's mean to increase the affinity of the antibody for antigens and the switching to most immunoglobulin classes. Other than, we've got the IgM, and depends on the interaction the antigen stimulated B cells with the helper cells, and then the other cells and peripheral lymphoid organs, and these antibodies induce the microbial antigens alone tend to have lower affinity and to be less functionally, and then those induced with the T helper cell. So you know now the interactions between the B cells and the T helper cells is very easy. When you get the antigens will bind to the antibody, then send the signal to the B cells, express it on the MSC class 2. The MSC class 2 will bind with the T cell receptor. T cell receptor expressed on the T helper cell. Then the CD40 on the B cells interact with the CD40 ligands, and the co-receptor, which is called the CD4, will check the interactions, and then after that the T helper cell will release a lot of cytokines or different kind of cytokines, lead to activate it or to enhance the immune system. So basically here we've got the T cells and the B cells that when recognize the antigens within the same molecular complex in order to interact. So that's the here it's more like the more further details about the T cells interactions between the T cells and the B cells. An example here we've got the internal viral proteins. So basically here the B cells and here the T cells or T helper cells, and then the dendritic cells. So we've got too many ways to explain that. Basically, in the first picture here on the right panel, you've got that the B cells, it's recognized the surface epitopes of the virus can process and presented others antigen epitopes. So basically when there is the antigen or the antibody presented on the surface of the B cells and it has the B cell receptor as well, and which is tightly bounded to the antibody. So the variable regions of the antibody attached to the basically to the antigen, the epitope of the antigen, and the antigen here it's virus, that will get inside the B cells and lead to the like the lead to the which is called the hypersomatic mutation, send the signals to the B cells. In the next on the left panels, you've got the picture of the dendritic cells here, which is the antigen presenting cells, but it's a part well, it can be like the connect between innate immunity and adaptive immunity, the dendritic cells, but the B cell is a part of the adaptive immunity, and it is called the antigen presenting cell as well. So basically here, when the dendritic cells, sorry, recognize the pathogens on the cell surface, or the like the virus, that will lead it to distract it and destroy it, the viral, in its small pieces. After that will express it on the MSC class 2 here, and then will bind with the T cell receptor, which is the T helper cells, which is called the CD4. Here the T cell receptors will bind with the antigen expressed on the MSC class 2. And then the next to the CD4 T cells, we've got non-specific CD4 T cells, and the CD4 T cells, it has an inactive form and it hasn't been activated yet. In the downstairs, we've got here in the down, you've got here the T helper cells, you've got the T cell receptor, this is the MSC class 2 expressed on the B cells. So that's mean if we're gonna to combine the both picture, here the first thing, when the B cell receptor, that mean it's considered as the antigen presenting cells, it binds to the fragments peptides or to the pathogens, to the variable region, and then the variable regions will send the signal to inside the B cells. That will lead to XL, which is loaded the MSC class 2, the fragments or the particles or concerning particles on the cell surface that will bind to the T cell receptor on the T helper cells. This is the first signals will lead it to engage the interactions between the B cells and the T helper cells. We've got another marker expressed on the B cells. It's called the CD40, that's interacted with the CD40L on the surface of the T helper cells. Basically, and here that will lead to, and we've got the T cell receptor as well, the CD4, I mentioned that in the previous slide. That will lead it to enhance or release a lot of the antibodies. These antibodies will attach to the microorganisms and lead to then the called the macrophage to induce the opsonizations and lead to destroy the cells. So this is the diagrams. I think it's very clear here to show you how can the interact between the B cells and the T cells. So it's very simple. Here the T helper cells, okay, and here the B cells, the antigens bound to the antibody. The antibody will the antigens get inside the B cells, then express it on the MSC class 2, express on the B cells. Why it's MSC class 2, it's not MSC class 1? Because the B cell, it's a kind of the antigen presenting cells, and the antigen presenting cells, it's just has the MSC class 1 exists, the MSC class 2 exists on the antigen presenting cells that interact with the T cell receptor. And then we've got the another marker, which is the CD40 expressed on the B cells, which bind to the CD40 ligands on the T helper cells. These interactions will lead to release a more or variety of the antibodies that will take the pathogens and then enhance which is called the opsonizations. So here we go to the further or more details about the engagement between the T cell and the B cells. Okay, here I wanted to describe it, the B cells and the basically the B cells and the T cells interactions with the dendritic cells and the pathogens expressed on the B cells. We've got the A and we've got the B. So that's it looks like the summary between between the B cells and the T cells. Like a summary, the T cells, it's divided into types of the cells, into measures type. T cell cytotoxic CD8 and the T helper cell CD4, and both of them and the T helper cells express the MSC class 2 on the cell surface. Okay, the MSC, it's binds to MSC class 2, but the T cytotoxic T cells, it's bound to the MSC class 1. The MSC class 2 expressed on the antigenic presenting cells, which are the dendritic cells, macrophage, or the B cells. And the T cells, it has the T cell receptor on the cell surface. So when we get the infected with any kind of the pathogen, there is the first line defense of the immune systems will start to activate it. That's engulf the microorganisms and get it into the cells. That will lead to distract it into small pieces, and then when it instructed in a longer process, that will lead to express it on the MSC class 2 if it was the T helper cells. If it was the basically the T cytotoxic cells, so it should be the MSC class 1, because the MSC class 1 is expressed on all the nucleated cells, and the MSC class 1 basically it's because it's expressed on all the nucleated cells, so that will lead to express or loaded on the cell surface the virus or the cancer infected, and then will lead it to bind to the T cell receptor. After the bind to the T cell receptors, because if it was either the helper cells or the T cells are activated by recognition of the antigen presenting by the antigen presenting cells, and the antigen presenting cells, as I mentioned, such as dendritic or macrophage, have to process antigen presented them and the cell membrane along with the measure histocompatibility too. And basically with that lead to the clonal expansions, may lead to proliferations and differentiation of the basically of the T helper cells, of the T cytotoxic cells. That is means to lead to the to call the macrophage as well to engulf the foreign antibodies. In A, sorry, I've been talking about the T cells with the pathogens. In B, we've got the antibodies or immunoglobulins with the B cell receptor that will attach it to the microorganisms, because the B cell receptor is as the immunoglobulin would express it on the cell surface of the B cells, which is bound to the basically to the bacteria or any kind of the microorganisms that will lead it to get inside the B cells. That will lead it to the somatic hypermutation or class switching of the immunoglobulin, because the first immunoglobulin expressed on the B cells, it is called the IgM, and the IgM, so if it is the IgM, it doesn't match with the bacteria or with the pathogen, that will come inside the cells and send the signals to the B cells and say it doesn't match me, so the B cells will release another kind of the microorganisms will match with this kind of the microorganisms. This process is called the somatic hypermutations, and after that, when then they're activated, that will lead to the clonal expansions. It means to release the plasma cells, and the plasma cells will release a lot of the which is called the antibodies. these antibodies, it lead to the protect the body, and then basically lead to the to activate the immune systems. So if it's want to uh, uh, basically, so this is the diagrams about the between the or about the T cells and B cells, like the summary, because you know the T cells, or if it, let's get examples about the T helper cells activated by the antigens present in the antigen presenting cells, and they stimulated the B cells to produce it the a lots of the antibodies. So the two types of immune response generated by the immune systems of the body are a non-specific and a specific immunity. So you know, we've got the physical barrier and normal flora, chemicals barrier, phagocytosis, histamine, inflammation, fever, complement proteins, and we've got the complement of the non-specific immune response, and we've got the humoral and the cell-mediated immunity. All two components are specific. While basic of a specific immune response, the humoral immunity is generated by circulating antibody, and the cell-mediated immunity is generated by the antigen specific T cells. So basically now you've got the good idea about the innate immunity, and then the adaptive immunity, and you know the T cells with the measure histocompatibility, and the B cells and the antibodies. Now you go to the general idea about the immune system, how does it work properly. I know the topic, it's a bit tricky and it's very hard to get it, but you need to read it basically a lot of times. I will explain everything, so it would be difficult to you, so in the classroom, and hopefully you get a lot of information about the immunology right now. So because we're gonna start another topic next week with a new topic. So okay, keep safe and bye.